So what we've got, I think everybody has a pretty good understanding of, all right, so what DNA is. Your DNA quizzes are actually in PowerSchool, okay, the ones you just took. We have to look at those questions and see what folks miss most often, okay? So I'm going to pull that stuff up, and we'll look at that before your test on Tuesday. You have RNA, transcription, you have translation protein. You guys just practiced it several times. So I think we're good with that, mutations. But what we need to know too is how we can screw around with DNA, okay? So how do we manipulate or screw around with somebody's DNA? And there's different ways, okay? So what we're going to do is we're going to make a column of several ways, and then we're going to do a description beside each. And then what you guys are going to do is show examples in your presentation, plus what are the benefits and the drawbacks, okay? So this is what I mean. So we're going to leave, maybe leave a couple lines between each one of these, and then we'll just give a description of each, okay? All right, so the first one is going to be, it's called DNA. Making GMOs, okay? So here's our first one. All right, so making GMOs. So DNA technology is also called genetic engineering. Okay, so if you are going to be a group that's going to research this, there's actually a couple different ways we can do that. You're going to look up genetic engineering, and then you're either going to do it with making drugs like insulin and growth hormone and things like that, or you're going to look up how we do this to animals and plants, okay? And you'll see some examples of this. All right, so hold up. So genetic engineering is inserting another organism's DNA into another. Oh. All right, so here's an example. An example of this is how we get insulin for people who are diabetics. Okay, so if you know anybody that's insulin dependent, okay, so what they do is the little vials of insulin doesn't come out of some human's pancreas. What they've done is they've done this. Yep. So they take a bacterium's DNA and they cut it and they insert a human making insulin gene into that bacterium. And every time that bacterium reproduces, it makes insulin. And then they suck the insulin out of it and they give it to the drug companies. Okay, and then they get it out to the pharmacies, get it out to the patients. So your insulin actually was made by bacteria. Okay, another example, and you guys are going to explore some different ones, but another example is how we can actually make different animals glow, like the glowfish that you see at Walmart or at PetSmart. They have like a funky little glow to them, and what they've done is, as a little fertilized egg, they've gone in with a little tiny pipette and some enzymes, and they cut their DNA open, and they've inserted a glow gene from a jellyfish into that fertilized egg, into that DNA, and every time it reproduces now, every cell has jellyfish glow genes in it. And then eventually the whole fish will glow.
I don't think that's
For real?
Yeah, I don't think that's right either.
But that's what they've done. So some states will actually outlaw some of those animals. So if we can do it with the fish, they've done it to other animals, and you're going to see this on this little video clip. Okay, we won't see the whole thing, but I want to fast forward so you can see some of those things. So
I'm cold.
a transgenic
She don't make me young too.
What?
organism is made. All right, that is another word for a GMO, genetically modified organism. Most of the food we eat has been genetically modified. That means that if you're going to grow corn, then most likely on large farms, the seeds that they plant to make the corn plant, they've actually been modified to maybe be pest resistant, or maybe for them to grow taller and bigger than some other variations. They've messed around enough to where they've created different types of different variations. So it's also called, that's what it, genetically modified organism. Okay, so that's just another nickname. A transgenic is a genetically modified organism. Okay.
It's not like the cancer. It's been genetically modified.
Right. So one of the best places to buy food in Lexington right now is Aldi, because Aldi will, they actually are starting to restrict certain, selling certain foods because they're genetically modified. The problem is maybe it won't hurt us, but it might, because the FDA is not responsible for telling us what type food we have is genetically modified. They don't have to declare that. So, for example, you're going to see a little clip that talks about how they take a gene from a fish that lives in really cold water, insert that gene into a strawberry plant, so the strawberries can grow in cold places. So you're eating strawberries and they may have fish DNA in it and you have no clue.
Oh.
They don't have to tell you that. So you don't know. But you know what? They say that you don't,
If I put a DNA fish in me, will I be able to go into cold places?
Oh my God.
you might be, as not now, but if you, as a little zygote or as a little embryo, if we could go in and manipulate your DNA in those cells, then they would insert that foreign DNA into your genome and you could possibly have those characteristics. Maybe to glow, to be a little bit more resistant to cold. That's what the theory would be.
I'm gonna time travel back in time and tell my mama that.
All right.
Do that.
So here are the research topics.
What?
Think about which one you want to do because each group will have a different one. Okay, so it's going to be either genetic engineering.
You write that down too?
I mean, it's up to you because these are just the topics, so that way you can think about if this is the one you may want to do.
Are we-
engineering.
All right, there's going to be a couple. One is making insulin,
Genetic engineering.
like how we actually make insulin and other drugs.
Are we starting it today?
That's why you have the computers. Yep.
We're starting a project.
Okay. Yep.
All
All right. The next one is also genetic engineering, but it's of plants.
I'm gonna do the drugs. Just kidding.
And then the other one is genetic engineering of animals. So that's 3 different topics that will have 3 different groups. And the slideshows are pretty short. All you're going to do is tell us some examples, tell us some benefits and drawbacks. That's really all you're going to do. And you're going to try to find a really good little short video clip that kind of helps explain it or maybe show you some examples. Okay, yes, ma'am.
Could we state like the brief opinion?
Oh, and that's part of it. That could go into the drawbacks. Yeah. Or benefits, both. Absolutely. All right, now let's switch gears. Forensics. When you hear the word forensics, what do you think of?
Science.
All right, so what's forensic science? Okay, so what kind, you may use these kind of fingerprints, but you can also
DNA.
use what other kind of fingerprints? DNA fingerprints.
Oh,
All right, so this is forensic science.
I want to do making insulin and drugs. Can I call it yet?
Not yet, but you may have dibs. Okay, so forensic science. This is making DNA fingerprints. Not fingerprints from your actual fingers, but the fingerprints of your DNA, your genome, from some type of evidence. It might be from a hair. It might be from skin cells or white blood cells that's in blood
I would
that's
do
been left
that
behind.
So like the things you leave behind.
So it's these things right here.
Such
So-
as
What is that?
So everybody has,
protein.
these are actually little genes,
Oh.
and everybody has their own unique, unless you're an identical twin, you have your own unique sets of genes
I'm gonna take a put.
that are
I wish I
isolated in
had
a DNA fingerprint. So this is by gel electrophoresis. This is just how it's. Okay, so this is the process. This is what you're actually going to tell us examples of. What are some pros? What are some cons of actually? Yeah, so this is the actual process, and I'll give you actually description and all of that. So just, yeah. Okay.
Electrophoresis.
Yep, and just skip a couple lines because I'll go back just because of time. I want you guys to have time to sign up for a topic and then kind of dive into it and figure out who your partner is, or if you're going to work by yourself. But if you have a partner, then one of you is going to start that slideshow and share it with you, with your partners. That way you can go ahead and make assignments. Okay, so here's a topic. So we have 4 topics so far. All right, the next one is cloning. Okay. So skip a little space and cloning. So this is actually making genetic copies of an organism. Okay, so this would be like cloning
Me.
sheep, or the first mammals cloned.
Cows.
Cloning.
I read an article about that way.
So cloning. Okay, so we're gonna make so this is making identical copies of an organism. It is illegal to make identical copies of humans. We cannot clone you, if we can't clone any people, but what we can do is clone other organisms. And we're going to talk about, well, and whoever signs up for this, find out,
I want to do that.
how it's done
my god.
and all the positives and the negatives of it.
I'm on other stuff. I'll be like, hey,
Okay.
bro.
All right, so we have cloning, and the next one is gene therapy.
Tell them to watch it.
All right, and gene therapy is kind of new. It doesn't work extremely well yet, but and for some diseases, it's going to work better than others. This is where somebody has a genetic disease and you try to fix it by giving them the correct gene. Fix the mutation. The medicine tries to fix the mutation, so that way you don't have the disease anymore. Okay, so this is introducing a normal gene to a person who has a genetic disease. So it's a treatment, but you're trying to fix a genetic disease you were born with, or that person was born with. Okay.
We do. We got it.
All right. So let's see. So we have 3 genetic engineerings. We have making DNA fingerprints. We have cloning. We have gene therapy. And I feel like I'm missing one. Let me see. Oh my gosh.
Oh my god.
I think that's it, actually.
He's over here wanting to fight Peekaboo.
All right. So topics. So we need to sign up. Tell me who I need to see who your partner is. There'll be some repeats. Some of you will have the same topic just because if you opt to work by yourself. Okay.
I want making insulin and drugs.
All right. Cloning and cloning. All
I
right.
want to clone too.
Cloning's gone.
Also, I'm working with Josh
Insulin.
and drugs.
I got Ginger.
Gene.
I got Ginger.
Oh, yes, we got this other.
No, drugs.
No,
don't pass me.
Did you mark this gene therapy?
Drugs.
Did you need therapy?
Only have
Me?
That's my presentation.
Girl, boss.
Where about?
no. Why don't y'all do
Sweet.
you want to do gene therapy?
Yep, it's me.
Yeah, I have you. Did you want gene therapy or do you want
Yeah.
Salvador.
We both need. Yeah.
or do we have this one?
I don't know what's your last name.
I want gene therapy.
Salvador.
That's easier.
Mendoza.
Oh, I was gonna say, shoot.
Okay.
Salvador
I'm more
Mendoza.
interested.
So we doing
All right.
therapy? What is that?
And then let's see who's left. Are y'all partners? All right.
Oh.
Y'all do gel electrophoresis, the forensics. Okay.
Get on your knees.
All right. So y'all are good. If you already have a topic, go ahead and create your slideshow. You can do it Prezi or slides and share it with your partner. All right, Michelle, who's your partner? Okay, so y'all, you're good. Sorry. Good. Y'all
Wait, where
good.
are we doing this
Marcus, y'all are good. Lindsay, Latrice, what do you guys have?
That's not even biology.
Which one?
What are we doing it on?
Hold up. We don't have anybody. We don't have anybody on genetic engineer of plants. Well, I can give that to somebody that's not here.
You gotta check.
How many is
Yeah, that's the only one we're missing.
What's that about?
Where we alter the genome of crops and try to make the crops more resistant, like to pests or disease resistant. But y'all have a good one. You have a good one.
Are we supposed to do it on Google Slides?
You can do it back or
I got
present.
hit.
Ooh, I like present.
I got a question. It's not mine. Where do you go talk about the attendance? I've been marked absent for their period and I'm here.
You tell your teacher, and they're supposed to email the fix. But you can also go to Miss Glass. Miss Glass.
Yeah, I got left absent. The period. I'm here.
Present.
Yeah.
Copy skip is
I
still.
guess. I guess.
I was supposed to have 7 times.
Doesn't do what you do either.
That's not me to me.
Yeah, you can do that.
Okay,